I would like to introduce another presenter, Dr. Aleksandr Stakheev, the third presenter. He's a senior researcher from the Institute of Bioorganic Chemistry, and
not senior researcher,
not
just
senior
researcher scientist.
Okay.
Thank you. I'm from Shemyakin-Ovchinnikov Institute of Bioorganic Chemistry, and my presentation is about the use of molecular technologies for identification of mycotoxin producers and their application to decrease mycotoxin contamination in food and feed. As you know, mycotoxins are secondary metabolites of fungi of different taxonomic groups. First of all, Fusarium. This is my main object of study. Aspergillus, including aflatoxins, which are considered as the most potent natural carcinogen, and other mycotoxins like ochratoxins produced by penicillium, eniatins, and so on. I have to say that mycotoxin production ability to produce mycotoxins is considered as a serious evolutionary advantage, which acquired by different fungal taxa, and the ability to produce mycotoxins is not necessary to base process of base metabolism or base process housekeeping process, but it is very important in terms of conquering new ecological niches to infect different plants, new host plants. And here is the phylogenetic tree, which shows the correspondence between evolution of Fusarium genus of different taxa, of different groups of species and groups of species of Fusarium, and acquiring ability to produce different mycotoxins and other secondary metabolites as gibberellins. What is important from genetic point of view? The genes responsible for mycotoxin production are organized in clusters. This type of organization facilitates regulation of mycotoxin biosynthesis, and from our point of view, in terms of fighting mycotoxin contamination, it facilitates the choosing of target genes to make different effects on this on mycotoxin biosynthesis. What we wanted to make, what was our goal to, what was and has been our goal to study. We use different molecular approaches to different goals in field of fighting mycotoxin and mycotoxin producers. We developed a system of molecular identification and taxonomic study of mycotoxigenic fungi. We investigated molecular basis of mycotoxin production. We developed molecular systems, qPCR-based systems, to estimate fungal contamination of agricultural, of grain, of food, feed. And we have been developing an efficient and safe approaches to decrease mycotoxin content. We developed a system of new molecular markers for phylogenetic studies and taxonomy of Fusarium. Some genes, some barcodes, such as translation elongation factor alpha gene or beta-tubule gene, have been intensively used for Fusarium taxonomy and identification, but not always these markers not always allow to study intraspecific relations in some Fusarium taxa, like complex species complexes like Fusarium graminearum, Fusarium exosporum, Fusarium venaceum. We propose two novel markers, phosphate permease gene, PHO, and minichromosome maintenance protein. These markers have been used in studies of fungal taxonomy, but for Fusarium, the use of these markers are restricted. We sequenced more than 300 sequences of these genes of different Fusarium species from all Russian collections. This approach allowed us to verify taxonomic identification of collection strains, collection isolates, and I have to say that we concluded that there were a lot of mistakes in identification of collection species. And we investigated the interspecific polymorphism of trichotillin and iniatin producing species of Fusarium. We estimated the genetic basis of toxigenic potential of this species and discovered new subclades, subgroups of Fusarium species, such as Fusarium venaceum 5 cluster on the right dendrogram, which represents a new subgroup in Fusarium venaceum species complex. During these studies, we, for the first time, detected a newly described species Fusarium caffeatum in Russia. This is the first detection of Fusarium caffeatum in Russia. This species belongs to Fusarium equisating incarnatum species complex, and what is most important thing of this study is that Fusarium caffeatum is able to produce fumonizin biodine on rice media. Why it is so interesting? It's so interesting because species of Fusarium incarnatum equisating species complex usually do not have an ability to produce fumonizin biodine. They do not have a fumonizin cluster on their genome. Preliminary studies by PCR and by standard markers show that this strain contains some of genes of fumonizin cluster, but now it is under NGS analysis. We are trying to sequence the genome of this strain of the species. Fusarium caffeatum genome has not been sequenced, and what is most interesting, we are trying to find the architecture with fumanizin gene cluster and trying to understand how it is sequenced in this strain, in this species. Also, we have developed novel kits for high-specific qPCR detection of Fusarium. Here you can see results obtained by the use of a system specific to Fusarium graminearum. These systems are highly specific and highly sensitive. They're able to detect 10 or 100 copies of fungal genome in reaction. These systems are successfully used in our joint studies with colleagues from the Dokучаева Institute. For example, to estimate Fusarium contamination in soil, and they also can be used to standard diagnosis of Fusarium contamination in grain food feed. Which approaches are used to decrease mycotoxin contamination of plants, grain, or food? Crop rotation, resistant cultivars, application of fungicides. All these approaches have some restrictions in use. They are effective, but have some restrictions. In our work, we are trying to use safe, modern and safe approaches, like using of natural derived compounds, compounds of plant or fungal or microbial origin, which are able to specifically inhibit mycotoxin biosynthesis. And now we started a study of using of modern molecular techniques, RNA interference for mycotoxin gene silencing. As an example of the results we obtained, a compound produced by Penicillium centrinum fungi, compactin. We show that this compound is able to inhibit biosynthesis of aflatoxin biodine and deoxynivalenol and zearalenone, aflatoxin Fusarium Aspergillus flavus and fusariotoxins, DON and ZEN. We also try to elucidate molecular mechanisms of action. This is not exact mechanisms, this possible mechanism of action of this compound. We show that compactin treatment of culture of toxin-producing strain of Fusarium culmorum led to inhibition of expression of some genes of trichotoxin cluster. Regulatory factors which regulate transcription of genes of cluster, as well as enzyme coding genes. The only gene which was upregulated in this work was Tri15 gene. It is a regulatory factor, but its upregulation of its expression have a negative effect on the expression of other genes of trichotoxin cluster. And also we just started study of RNA interference to inhibit mycotoxin biosynthesis for Fusarium and Aspergillus, which we have chosen
set of genes which will be tried to inhibit to be silenced by RNA interference. And we are going to use different approaches of delivering of RNA to target cell, like host-induced and spray-induced applications. Here is a list of key publications of last years. Thank you.
Dear Aleksandr, thank you very much for your presentation. Maybe someone has questions.
Thank you for your presentation. Could you tell me, please, what is the ecological role of mycotoxin production in environmental condition?
The main role of mycotoxins is mycotoxins. Some mycotoxins, not all mycotoxins, are virulence factors of fungal to host plant. The ability to produce mycotoxins facilitates the infection process of fungi to host plant. This facilitates growth and development of fungus on the plant.
Well, thank you.
Thank you. Maybe some other questions you have?
You just introduced that RNA interference. You have developed some method to use this method to control disease.
Could you please repeat, maybe?
Or maybe you have some preparation, or we call the biopesticides. Use this RNA interference. This is technical.
Could you please maybe tell a little bit more? I mean, because I'm not sure that I understood the question.
Yeah, because we use the RNA interference interference. This technical is useful for the control the pathogen, like the fungal pathogen. Also, mainly is the fungal pathogen. My question is, you already developed some the pesticide, the RNA interference pesticide, to use in the practice in the agriculture.
We are doing that. We are doing that because I have to mention that this project started as a project of to increase the quality of potato plants. Yes, and these techniques, including delivering of RNA to cell, of spray techniques, were developed to potatoes. And we now will be trying to apply these approaches to fungal species. Firstly in vitro and then in planta. We will be trying to do that because actually, in the last 5-10 years, some approaches based on RNA interference have been developed
That's very interesting.
Yes. To fight mycotoxin contamination. But not all these approaches came to in planta studies. Many of them are many of them were good in vitro, but not of them. We will try to reach in planta and estimate the efficiency of this approach.
I see. Thank you.